You know what's so funny is I told Josh about this. I was like, Jeremy's being mean to me. And he was just like, good. And I'm like, whose side are you on? Your wife's side or his side? And he's like, Jeremy's side. I was like, good. I don't like this.
There you go. There's that. Then when you do get in, you'll just click in, and this is your clicker. Should you want a clicker?
Should I want a clicker?
Should you want a clicker? Oh, Sean already with the essay in here. All right. Well, that was a comment. Where's Kenzie?
Her and Elise are finishing up a behavior for a group, which is like, she knows how to do this. I don't know how to do with notes.
Casey probably knows how.
Casey, how do you get presenter view on where like I can see my notes?
All right. This will be good though, because remember, you guys have a symposium at the end, such that you're going to have to do the same thing.
Do I need to download it?
Yeah, downloading it's the most easiest way.
So this will kind of be
two screens up, because then when you hit present, it automatically puts it in present.
what you're doing.
She's got to download it.
I'm gonna download some malware to your laptop.
That's fine. I know how to clean it. It's not that big a deal.
I don't know whether I should be concerned or impressed with that. Kenzie! The pigeon is freaking out.
Are you pigeon?
Yeah.
You said the pigeon is freaking out.
Yeah. Are you figuring it out?
No.
I think if you just hit present now, you're good.
That one works for you. Oh, you gotta. There you go.
Oh, no, it worked for a second. Yeah.
There you go.
Okay.
Will you hand me my beverage?
And then forward, backward, laser if you want laser. It's a green laser.
It's a green laser.
It's a green laser.
Does it actually work on? Oh, it does work on that.
It makes it easier to see because it's green. All right.
But people have green color blindness, though.
Well, it's still a light. It's just gonna. It should still be easier to see.
People. Okay.
Savannah, you can sit here or there. With Carl? Okay. All right. Welcome, Abby.
Hi,
again,
everyone.
graduate student out of Dee's lab, getting ready for her
I'm getting a full-on
thesis
intro.
proposal. And feel free
No.
to make her feel welcome, hopefully. And then as she's going through it, give her all the constructive feedback.
Key word there, Jeremy. Constructive.
Respectful, but brutal at the same time. Got
I
it.
think he doesn't like me because I cut his hand. So
Spilled a bunch of chemical and then
spilled a bunch
broke
of
glass.
chemicals. It was a rough day. Yeah. That was fun. That was a fun Friday. Okay, so I have two potential title ideas which you guys can vote on or not care about. Completely up to you. But the main one that we're kind of going for right now is parental care across embryo development and experience in newly transitioned males in Lythropnus dolly, also called the blue banded goby. All right, so give a general overview here. There's a couple different types of parenting. The first one up is biparental care, also known as alloparenting. A really good example of this is called the zebra finch. So typically they are socially monogamous creatures. They like to hang out in pairs, raise their chicks together, things like that. You have California mice who raise their pups together as well. And then another great example is us, humans. And then going into uniparental care, so it's just one parent that does all the care. One example of that is elephants. Another one would be orcas. They're really good moms. And then obviously everyone's heard the expression, don't mess with mama bear. Bears are a great example of maternal uniparental care as well. And then finally, we have paternal care, another form of uniparental care. So we have seahorses. Seahorses carry their young in their bellies until they hatch out, and then that parental care is over with. And then we have the giant water bug. These guys are really cool. So females will lay their eggs on their backs, and then they will carry them around for 2 weeks until they hatch out. So this is a photo that another professor here at ISU graciously like sent to me, and this is what they kind of look like after they've hatched, and they'll stay right there as well. And then we have the blue-banded goby, which is the primary example that we are going to be focusing on today. So a little bit of background on the blue-banded goby. So they typically, where we get them is off the coast of Catalina Island in California, and they typically will live amongst like sea urchins, rocks, any sort of like nooks and crannies that they can kind of hide to raise their youngs. All the dads do all the parenting behavior, so they do this thing called fanning and rubbing behavior. So they'll kind of like press their bellies on their eggs and do like a little waggle and everything. And what's also great about these fish is that they can switch from female to male and male to female. So they're bidirectionally hermaphroditic. They can do that as many times as they want in their life if their social environment requires them. So if there's a bunch of females in a rock area, one of them that's typically the most dominant will then transition into that new male and start raising and parenting the next round of embryos. So that's a nice, cute little male. Gravid female, you can see the big belly of her ovaries. And then for our project, how we simulate a nest environment in our lab is we basically just take PVC tube, put some like clear plastic around the edge, and then they just have their eggs like this in there. So yeah, that's how we kind of simulate that. This is a nice, terrible photo of some orange embryos lined the edge of that as well. So for my study specifically, we're going to be focusing on protogynous sex change, so female to male sex change, whereas protandrous is female to male. So just focusing on protogynous for this one. So this is what the female's ovaries typically look like. Pretty standard, things like that, to then basically having regressed ovaries, starting to maintain some sort of nest territory, defending that as well, but not able to produce like probably any sperm yet, to then being fully male. You can see how different their gonads change to actually being able to produce sperm and fertilize embryos as well. And then another thing that we can use with these fish is external genitalia. So they have something called a genital papilla, or GP. And that's a really nice, easy way for us to kind of look at them without having to dissect them to figure out if they are male or female or transitioning, which is really nice and not as invasive, obviously. So this kind of like rounded shape is a really good example of a female genital papilla. Then we go into transitioning, so it's starting to get a little bit longer, a little bit more pointy, and then into a male where it's completely pointed, looking a lot different from the female, fully able to start producing sperm as well. Okay, so give me a lot of words on this one, so I'm sorry in advance. But my general hypothesis for this study is that newly transitioned males, which we'll call them new males, will have distinct patterns of gene expression and parental behaviors compared to stable males and then stable females who don't parent at all. And then we're also going to be tracking embryonic development in this study to reveal how good a parent a fish might be as those embryos develop along with their parenting behaviors as well. So I got this idea from a real famous scientist, obviously, Howard Wolowitz, where kids are like pancakes. The first one is always the throwaway. As an only child, I understand that fully. Where maybe if you're a first-time parent, you're probably not going to be as good as a several-time dad that's done this multiple times and goes, I know what they're doing, things like that. So the general theme of it. So aim one for this first study that I was is to determine how tissue-specific expression of steroid metabolizing enzymes and receptors differ from those new males, inexperienced dads, to experienced fathers caring for late stage embryos. And what are late stage embryos? This is embryos. So I have taken photos tracking embryos and their life cycle. They typically hatch within 4 to 6 days, depending on the tank water and temperature as well. For specifically late stage, we're going to be looking at right in here, around that stage. So they're typically anywhere from 36 to about 2 hours away from hatching. Just kind of depends on when they lay and when we are able to take that group down, things like that as well. So really confusing diagram here, but these are some of the genes and enzymes that I'm working with. So for 11-beta HSD, this is really good and important in stress regulation. So that kind of helps deactivate cortisol, which everyone kind of knows it as the stress hormone, and converting it into cortisosterone, and kind of protecting other tissues and things like that from other stress signals that might occur. In reproduction, 11-beta HSD is really important for converting that into 11-KT, which is an important androgen for male fish in spermogenesis and things like that. And then we have AR down here at the bottom. And AR kind of works hand in hand with CYP-A1A and A1B and kind of metabolizes those androgens, any sort of estrogens in the body, which can kind of help balance out those things, and is also important any sort of aggressive behavior as well. And then you'll notice for the CYP-A1A and A1B, basically this is just another way of saying aromatase. It's just the version of aromatase in these fish. A1A is for gonad specific expression and A1B is for brain specific expression. So since we're looking at gonads, GP, and brain tissue in these fish, we gotta have to have both to look at to kind of figure out what is going on. All right. You can bully me about this slide. I don't like this slide either. But for this, I'm going to be looking at gene expression. So basically, this is just showing the different dissection pieces that we get from a fish. Yes, the brains are really that tiny. And basically what we do is we homogenize them, extract RNA from them, and basically run a plate looking at specific genes of interest. And what's really difficult about these fish in particular is that we don't have a full sequence genome of these fish. So every sort of primer that I build off of to try and like figure out where in their genome that AR, anything like that is expressed, I have to use reference animals to basically develop primers that are basically characterized for those specific fish. So it takes a little bit of troubleshooting and things like that, but thankfully we have NCBI, which is a program I use to kind of help build that out and use references to compare, so I'm not counting all the ACs, T's, and G's by myself, because that would not go over well. All right, and then predictions for this study. We predict that new males will show reduced nest attendance and embryo-directed behavior, so those waggles and rubs, when compared to experienced males who will probably invest more time into nest care. The expression of CYP-A1A and A1B will be elevated in reproductive tissues in the gonad and genital papilla, so that external genitalia, of late-stage carrying males, suggesting that local steroid hormones and regulation of reproductive physiology during parenting. And then finally, new males will also show, try the same one twice. I do, that was my bad. I didn't copy that in right, whoops. But anyway, so some preliminary results. So again, we have our GP photos kind of showing the difference between a female and a male. We took images of our fish, like right when we processed them, to put them into groups, and then afterwards to see how that GP would change. You can come in, Ashley. And we did see significance within that they were getting more male-like as well, and very significant p-values. So it's great. They're changing. They're actually able to produce sperm, actually fertilize the embryos that they're taking care of, things like that as well. So it's a good verification. And then next, we have waggle time percentage. So we watch behavior for 10 minutes on these fish before we take down that group. So at most, no one waggled more than 15% of the time in a 10-minute behavioral period. So that's important to note on the y-axis there. On the x-axis, we have stable females. Females don't parent in these animals, so we shouldn't be seeing any waggle time from them. So that's great. And then we saw no difference between new males and stable males. So far my prediction is wrong. They're actually parenting just as well, which is cool. And then moving on, we have time in nest. So if they spent 100% of the time in the nest, that means they spent that entire 10-minute behavioral period in the nest with their embryos. So actually there was a difference in this between the new males and stable males. Sorry, this is kind of covering it up a little bit, but stable males are right there, which is really cool. So maybe not in like the directed care that they're giving the eggs, but they're at least defending their territory a little bit more than a stable male might be, because he might be a little bit more secure. He's been a stable male for a long time. He knows what he's doing, things like that. So yeah. Alrighty. And then going into my second aim, which is to describe how 2 stages of embryonic development shape parental behavior and gene expression, and determine how these relationships differ between newly transitioned and experienced fathers. And I forgot a point, sorry. And that is because what we saw here is that these 2 females spent 100% of the time in the nest tube. And I went back to the behavior sheets, they were spawning during this. So we did not control for embryonic development in our stable groups for this study. So we're like, hey, this is kind of, we should be seeing a significant difference between these 2, and we're not because of those 2 females. So we need to control for that for all of our groups, basically. So that's what kind of led us into the second study. Sorry about that. All right, so predictions for this one. The stage of embryonic development within nests should influence the amount of parental care exhibited. So when embryos get older, they will twitch and move and provide heat to each other. So maybe during earlier stage development, the male is in there a little bit more to make sure they're getting the heat and nutrients and cleaning that is needed for that. And then specifically, newly transitioned and experienced males, the new dads, will exhibit distinct tissue specific patterns in GR, 11-beta HD, and AR in the brain and gonad and caudal muscle compared to experienced stable males. And then because we're looking at glucocorticoid receptors, that's what GR is, we're also looking at cortisol measured in EIAs, thanks Jeremy, will differ in the brain and caudal muscle between new and experienced males, kind of showing that there's probably some sort of investment in parental status that's different between the new males and the stable males. So got our embryo photos again. So we talked about the 2 stages. So this one, we're doing early stage embryonic development. This happens within the first 24 hours of being fertilized. So you have to be really quick and on it to make sure that you're catching that stage. And then also we've shortened our parameters for late stage a little bit more. So that way they are only at most 12 hours away from hatching to kind of catch that like last bit of parental behavior before the embryos hatch. And then we got a couple different genes here. We're introducing glucocorticoid receptors. Everyone knows that cortisol is just a part of everyday life. If you have chronic, whoa, what did I do? See, Jeremy, I don't like your mouse. But GR is, if you're chronically stressed out, that it's going to have metabolic effects on your overall body as well. So that can affect reproductive growth, how much you spend in the nest, things like that as well. And like we talked about before, 11-beta HSD also inactivates cortisol. So that's also nice to look at the interplay between those 2 enzymes and receptors as well. All right, still doing qPCR, all the fun stuff with that. And then also we're going to be working on an EIA to just do cortisol because we cannot make primers for that using qPCR. And then preliminary data, still working on it. I have an N of 3 for some of my stuff. So as of right now, we are showing no significance for certain things. And one thing I love and hate about these fish is the individual variation is very high. So still working on that. And then this is my acknowledgements page. And for context, this was the day that Kenzie and I had accidentally planned 20 dissections, and it takes us about an hour and a half to dissect 2 fish. So you can do the math on how well that was gonna go. And that was also the day that someone came in and took photos of us. Woot woot. This is Catalina Island. This is our field site that we go to every summer to collect our fish. And I got most of my work done for this project during that. Yeah, that's all I have. Thank you, guys. Don't like this mouse.
Can't help but notice Casey's name's not on there. And my last name's spelled wrong.
Did it?
Starkly.
What? Because I didn't put Samuel? Okay, God only gave me, they only gave me my ability to spell my own last name. Okay, I'll fix it. This is the last thing I did. Don't let me do things at 2 a.m. All right.
And I know this is fish instead of mammals. I am aware of that.
Here, I'll put it back
However,
on.
it was, it's science. So you see how this is immediately applicable still, right? Okay.
Wait.
Questions / feedback?
There you go. We have the rat. That's a mammal.
Or, yeah. Okay.
And humans.
Yeah. And the polar bear.
Yeah. Well, the polar bear's on the next one.
Well, yeah, but he, she was there. Feedback? Questions? I'll have plenty for you later.
Oh, thank you so much. Yes.
How long was the embryo development?
4 to 6 days, depending on tank temperature. So if the tank temperature is on the colder end, like 17 degrees Celsius, it'll take closer to 6 days. If it's a little bit warmer, which it typically was in Catalina where we were at, because we went in the summer, that was around 20 days. We had a group fully develop to late stage in 4 days. So we gotta be a little flexible with stuff is what I learned. Yes.
What sort of sample size are you going for?
8 of each group. For my first study, I have an N of 10 and then an N of 9 for all the stable groups, so it's a little bit bigger. But yeah, for this next one, because I have so many groups that I'm comparing against, it's going to be closer to 8. Yeah. Yes.
So you mentioned you were recording the waggles and the buffs
Mhm.
in males. Does that just stimulate the embryos, or what is the benefit?
Yeah, so it does multiple things. So typically when they're rubbing, that could be them putting their sperm into the egg to have it be fertilized. What it also does, it kicks off any sort of debris that might be around them. It applies heat to them, which is really good, especially if they're in a colder temperature. It helps them develop a little bit quicker because the longer they sit there, the more prone they are to predation, things like that as well. And something that is also as a coming soon for my second project is the females like to cannibalize the eggs. So they like to sneak in there and go nibble, nibble, nibble. And unfortunately, with our new males, we have some cannibalistic groups as well, where the new males were eating their own eggs that they fertilized. And I was like, what are you doing? So there's a bunch of reasons for that. But yeah, it just helps clean them, make sure that they're fertilized, things like that as well, which is good.
Any others?
Yes.
Besides individual variation and now cannibalism, what other possible experimental errors are possible with these fish that other genetics studies?
Yeah, that's a really good question. I think just the fact that we don't have everything fully sequenced is a really big part of that. For a lot of genetic studies, especially if people work with the more common organism like zebrafishes, they've had annotative sequences 10 times around, reflecting on it, refining it, making that sequence bigger. Whereas I just have to do little chunks, basically. So if you did the entire sequence, you'd have a better outlook, you'd have a more specific result and things like that as well. And then a previous master's student that just recently graduated that did this as well, he saw a bunch of individual variation between that expression as well. So yeah, it's just individual variation with these
Due
fish.
to what?
They're mean. Well, they're outbred fish. They're not inbred like typical mice and things like that you see a lot in research and things like that. So just
What causes the variation?
being their own animal. Just like we all have individual variation within human studies because we're each our own human.
Okay, more on that later.
Fine. Yes.
How long have you been gathering this data
So the first study was done last summer and I wasn't even here yet. So that was actually kind of done for me, but all the primers designs and things like that I've done. And then I have Sam right here that's doing part of the qPCR work for the RNA extractions, which is really nice. And we're about, this is year 2 of collecting that data, basically. And then for my second study, I just started that this summer in Catalina. So this is like 8 months in, basically, to it. So yeah, takes a little bit.
Okay, cool. Well, thank you.
Yep.
I'll bother you later
Oh boy.
after class.
Yeah, I'm
Just
gonna leave.
escape. Just hit escape.
Can I take this thing off?
Yeah.
Where's the button?
It's on the side.
Which side, Jeremy?
It's there. Use your eyeballs.
I am.
Well. All right. Yeah, you walk by the keys.
Thanks, Jeremy.
Thank you. Well, that was fun for me. So we were making a buffer, and you have to make up, the buffer needs to be a certain molarity, and you make it in 1 liter sections, bottles. And so she went to set them down, and me just trusting her thought she set it on a stir plate. No, did not set it on a stir plate, set it on a shaker. And I turned it on and it just shook everywhere. Bottles hit each other. They're glass. And so it just shattered. And I went to grab it because it was like starting to fall over. It was going to shatter even more. And then it just sliced open my palm. So, you know, what a good time that was. All right. Walker. Charles Entertainment.
You know, I feel like an expert on the Blue Band Recovery. I sat in on McKinsey's master thesis last year too.
Did you?
And then Lena talked to me about the Yellowstone trip.
Just saw a nice large hobo spider on the ground. It was over here. Gage. Sean. Austin. Hallie. That was like perfect, right in your hands. Sarah. They do at some point. Yeah. All right. So for this, some of you, like Charles Entertainment Cheese in Brooklyn, will note that I whited out quite a bit on section 4 because when I made this, I read section 4 in my mind. I know that's not what it says. That's why I whited out a bunch or scribbled. In my mind, I read that as what are the 4 unique traits that mammals have, which is not at all what that question is asking you. It's asking you just shared by all mammals, which is hugely variable. And then as I was grading it, like the first, I don't know, 3 or 4, I was like, nope. And then I reread the question. I was like, let me go back and fix all those that I already scribbled out and did, right? So you'll notice that. That's a my bad. I have since changed it in the exam to write 4 unique traits that mammals have, which are hair, 3 inner ear bones, a dentary, right? And it was dentary, not mandible, right? Almost everyone wrote mandible. It's dentary. I remember like on one diagram it does say mandible, but like all the other ones it says dentary. I made a big deal about it being a dentary. P. Cook said it was a dentary. This is your dentary, right? Okay, the whole thing. A few of you put bison for genus. I understand why. If you did that, I just circled both bison and then went through a line. It was just, I mean, you could have just copied it down. It was right there for you. True and false, that was hit or miss. Some people did great, other people not so much. Some people did great there, not other places, so it was variable. The one that stood out to me that got a lot of people more so than the other ones was 36. The left ventricle pumps oxygen to the lungs. No, that's the right ventricle. The left ventricle pumps it to systemic circulation, right? Right ventricle is the lungs, left is to, yep.
So for
34. Yeah.
Didn't he not discover it? He just furthered ideas from his father.
His grandfather came up with the original concept of that. But in history and everything, it goes to who's credited for it, which is Charles Darwin. I mean, if I mark something wrong somewhere and it's right, I'm happy to change. I'm not going to be a stalwart person if I'm always right. All right, I'm not going to play that game. But history credits Charles Darwin, so Charles Darwin. Uh-huh. Oh, well, there's why I disagree with Dave.
It's not the concept. He's like, it's natural selection. Right.
Yeah.
Oh, the mechanism.
He did. Yeah, he discovered the mechanism by which it was. Yeah.
And so then I was like, okay, it's not the concept then. But
I could go back and forth on that then. If you got it wrong, let me know.
I got it wrong.
Well, okay. You're gonna have to email me or canvas me because I won't. I won't remember. Yeah. Slow it up. Was it a zebra, or because certain animals within Pestodactyla are like horse-like, or a horse is not a domestic horse.
Oh, you did specify.
Yeah. So like a zebra, yes. Yeah. Right. The other thing that struck me is many of you put 3 vagina for Metatherian. 2. No, it says 2 vaginas and pseudo-vaginal canal. What does pseudo mean? False. Pseudo-vaginal canal is where the 2 come together, but that itself is not the vagina. That's different.
Is one useful?
Read the thing again. Oh, I know, because you before you were circling all the letters and then you just wrote it in the blank. No, it's not. Is it? No, it's not. So I was just like, why are we suddenly changing how we're doing?
So the 2 vaginas, those 2 are for sperm storage, then the pseudo vaginal
It is like the birthing canal.
That's what I wrong, because I thought it was 3 vaginas.
Mm-mm. No. I think that's where people were getting 3 from. Was this when I went back and looked? It's like that's got to be it, right? Like, yeah. The bone diagram. How did you know 5 was the ulna? Because it's okay. Because your ulna if you look, it's the one that comes up and forms the olecranon process, or your elbow. That's how you know it's the ulna.
I just memorized.
The humerus sits in that joint.
Yeah, that's what
Yeah.
I was like. I was thinking like this doing this, but I was like, no, the dog's sticking.
Yeah,
Oh, really?
Yeah. Well, that's how it's spelled.
yeah.
I
This
just
is
remember
why
from the diagram that the old.
And this is anatomical position in A and P. Yeah. So
Also, I thought the ilium was higher.
Yeah.
the ilium is the giant bone that makes up the pelvic girdle. It's not just the top. It's not the whole thing either. So it's this right here. I'm just going to use a porcupine. This right here, all of this is the ilium, right? And then very down here, this is the pubis, this is the ischium. But this whole thing right here is the ilium.
Okay. It's pointing at the pelvis. Yeah, it points just right in the center. And so I was like, I was torn between. I said it's pointing to like the entire bone. It's not just.
Let's see.
Yeah.
I mean, that's kind of still up above it. I don't know about that one. No, that's the iliac crest, but the pubis doesn't start until down below where the acetabulum sits.
Also, further in my case, on this diagram, it does point to the ilium just like.
Let me walk my fat ass over here.
Because yeah, I mean, and that just misunderstanding myself, but yeah, I thought it was like just the dish.
Okay.
No.
Of it.
No, yeah, the ilium comes all the way down here.
Fair enough.
And then the pubis is right here below. The ischium's back there.
With 6 and 10, it threw me off. They were both metatarsals. Why?
Metacarpals for 6, 10 is metatarsal. Metacarpal, metatarsal. Yeah.
I'm just glad it is.
So while I was grading this, I wanted to be somewhat merciful in areas. So that's why I totally opened up section 4. Anything you wrote there that all mammals shared was fine. And then as far as like the short answers and stuff, I wanted to be somewhat open on those as well. At the same time, knowing this is a 4,000 level midterm, so there needs to be some level of good rigor that you need to adhere to, such that in female defense polygyny, for example, in elk, you got to tell me that it was a male that is in a harem. He's going to mate with multiple females, hence polygyny. You can't just say defending. What is he going to do for defending? He's going to mate guard. He's going to chase away other females of this nature, right? You have to say these things. As far as animal, you had to tell me, you at least had to like tell me a few of, it was a metazoan, it's heterotrophic. It can move at some point in time because it's a mammal. It's multicellular, right? Does it have to have a vertebrae to be an animal? No, a sponge is an animal, right? Gnat is an animal, right? So a vertebrae is not required. So marsupium, that was probably the easiest one out of there. You just had to tell me that was a pouch of a female metatherian that goes on for development, right? That was, as far as the dental formula, I think you guys pretty much crushed that one. That was good. I'm glad you guys did pretty well in there. The biggest mistake people made was they put 17 and then they told me total teeth was 17. Yeah, that was just one side of the mouth. So it was 17 times 2, 34. That was the total.
I knew that. I just misread
3
over 2 as 2 over 2.
Yeah.
So I was like, oh, well, that's
yeah, and that was probably a -1. Yeah. Which, I mean, it's worth 5, so that wasn't too bad. For the long answer, that one was, you had to say monotreme, right? Metatherian, eutherian, of course, right? Those are the 3 groups of mammals. Then to get all of the points for it, you had to, okay. some of you said metatherians give birth early, but that's ambiguous. Like, what do you mean by early? There has to be some kind of comparison between metatherian and eutherian. Earlier than meaning what? Like a couple people wrote very early such that they're blind and only have 5 digits and they crawl into the marsupium. Excellent. That's exactly correct, right? They don't have anything else developed. Perfect. That's exactly what it is. And then if you were to say eutherians give birth to fully developed young, that's true. But like at what amount? Someone then went on to explain altricial versus precocial. Excellent, because that's not the same thing, right? So that's where you got to know what these things are to get the full amount here, right? I try not to dock too much for certain things, but again, it's a rigorous course and you need to understand the terminology and what it is, right? That's required. Such that you could have a discussion with me or any of the other professors in the department, say, I know what a metatherian is. It's defined by these things. This is how they're different, right? And you can use these terms, throw them out. You can throw out what it is and have a I don't know, 5-10 minute conversation with them, right? All right. If the Charles Darwin thing, I can go back and forth on that, I guess. So I can be merciful there. You let me, you're going to have to write it to me though on Canvas or an email because I will forget. Yep.
Could you explain with number 75, the mandible versus the dentary?
Dentary is what it's referred to in every single mammal ever except for humans.
Yeah. On the thing that we're supposed to study.
Look at the slide after that, and the other, yeah, and the other one, yeah.
It is, but this one explicitly says memorize all.
Yeah, well,
I was told to bring that on.
that one you can make that argument, but I know we had like a 5-minute discussion about it being a dentary, so.
5 minutes over how long many hours of
I'm going to stay steadfast on that one.
No, don't wait, Walker.
I'm writing the email for the Darwin.
Darwin, yeah.
Copy me on
Depending on how you did, that could, if you're on the cusp, that could put you from an 89 to a 90, maybe.
Well, yeah, it's
Or-
85.
3 points each. That would be
We have a.
Because I remember that very.
No.
This one, regardless, is the best exam.
Is that good or bad? Does that saying something about the exam being too easy? Should I make the final harder? Mm. If I did what?
From giving the 3 points for number 75, it bring me to 80. Bring up the entire class.
No, that's not percent. It's just 3 points. There's no way it brings you that. 3 points is not that much. Yeah, no, it doesn't. I was like, what are you talking about? Can you do math? Okay. The overall score, I was told you can see the high, low, and average, but the average was 77, which is pretty good. I mean, that's what you'd expect. It's pretty good. And high was 94.
You got 94?
No, I got 100.
Except for the Charles Darwin.
You could argue that.
Could bring me to a 79.5.
Ooh, rough.
Well, no, I just made a comment on the midterm. Yeah, so did I. I even saved one.
Yeah, email me. That way I know I get a direct thing. And plus it gives you a record of it, right? So you can go back and say, yeah, but you want stuff for your own student records in case I do mess up somewhere. I'm not going to mess up on purpose, but you can just say, look, I emailed you here. Here's the receipt. Good, great. Protect yourself as well.
One more argument. Aren't all mammals synapsids? No.
Hair neocortex, complex skulls.
Yes.
I just went with your first answer.
Hmm?
I just went with your first answer.
Did I not lose points in this?
Well, complex skulls is they're actually less complex, so I went with that and just didn't even look at this
Oh, okay.
because you have 2 answers there.
Yeah. Well, I was talking about the skulls themselves. Yeah.
Yeah,
He's like in.
Okay.
more complex skulls are synaptic. Which one do you? Which one do you mean?
I meant synapses, but I was thinking
Okay,
more
but you have two answers there. Which has,
So you chose the wrong.
like, well, yeah, I chose the first one in the space. That one's on you, not me. Antarctica, there's seals. Leopard seals are down there. Mm-hmm. Yep.
That was what I could think, but I was like, I don't know if they like live there. If they just visit here.
Yeah, no, they're there. They're there permanently. They're the top predator in Antarctica as a leopard seal.
I did not put true on that one. The rhapsodies arose from synapses. Weren't they a divergent path? I know that one.
Yeah.
I've taken paleontology.
I think some people just selected some as they were going through and didn't reread or go back or something, which is common on an exam. It happens. There are some of them. I don't know how many of you know this. Probably at this point you're fully aware, but you know when you take a large exam like this, if you don't know, you just skip, and then there's probably the answer to the one question somewhere else in the exam, right? Like almost always that's the case. There's only so many ways you can ask certain questions in the same concept. So you just wait and see if it's answered in the previous or in a following question.
That's how I got my calculus.
Yeah, right. That's what you do. Yep.
So did the rhapsodies arise from a group that wasn't technically?
They're split. It's a different evolutionary pathway.
Anapsids then? They have a common ancestor?
Yes. Right? Yes. I believe that means nothing to me. Yeah. I wish I knew. That's more of a P. Cook question. Yeah. If P. Cook says I'm wrong, then I'm happy to, you know. Well, no, I'm happy to be wrong. I'm not going to play that game of I'm always right. I'm wrong constantly. It's fine. Like, you know, I mean, I feel like it's more respectful to just admit I'm wrong, and then we'll go with the right answer, and then that's the case, right? It's really frustrating to have someone be like, "No, I'm always right." And they do that. Like that's aggravating, right? I'm not going to play that game. All right.